Welcome to this video, which is part of a series of training videos for the Colonial Waterbird program. This video will cover the Gulls, Terns, and Shorebirds nesting stage guide. This video will include a protocol overview for the nesting stages, a nesting stage guide for gulls, terns, and shorebirds with species identification, descriptions, and photos, nest identification, additional information on subcolonies, and an example of incomplete and complete datasheets. The protocol for the goot species is 1. Visit the colony. Watch for courtship, nesting, and breeding behaviors. 2. Fill out the first datasheet, which will have the total number of adults, active nests, and chicks in the colony, and a space to write information about habitat changes and disturbances. 3. Fill out page 2 of the datasheet by tracking the nest stages and counting adults, nests, or chicks. 4. Finally, make a map and mark the subcolonies that you are tracking. The species you will be looking for when filling out these datasheets are American avocets, black-necked stilts, Forrester's terns, and Caspian terns. There are key characteristics to look for when identifying each species. American avocets are a large shorebird. They have long legs, a long upturned bill, black and white upper parts, and a rusty or gray neck and head. Black-necked stilts are large shorebirds. They have a black face, hind neck, and back, white throat and underparts, long, thin pink legs, and a long, thin, straight black bill. Forrester's terns are medium-sized terns. They have a black cap, the tail is long and deeply forked, the wings are white in most plumages, and the legs are orange and relatively long. Caspian terns are large, gull-like terns with a black cap, white body, a large, brilliant red bill with a dark tip. This photo has several different species. Sometimes the sites will consist of many species, so practicing identification or bringing a guidebook is helpful. How many species can you identify here? Here we have the black skimmer. It is not a primary species to survey for, but they can be around other primary species, and we do want to record their breeding. This is an American avocet. Here we can see the orange rusty color of the face and neck, and the upturned bill. This is the Forrester's tern. You can see the black cap and orange bill. Finally, we have the black-necked stilt hiding in the back. You can see the white throat and black bill. In addition to identifying species, you will also have to identify different stages of nesting. There are several behaviors and characteristics to look out for when you're in the field. At stage 0, the birds will be at a pre-nesting stage. You will see birds at or near the colony using displays of courtship rituals to attract a mate, such as dancing, building nests, mating, or roosting. Stage 1 will consist of egg-laying or incubation. The adults will likely be on a nest for a long period of time, or standing at a nest to turn eggs. It can be difficult to distinguish an incubating adult from a roosting one, so you may have to watch it a while to look for behavioral cues. Stage 2 will consist of small downy chicks with soft feathers. Adults will likely be at the colony, feeding them, or standing near them. Stage 3 will consist of larger chicks that have lost some of their down and are growing new feathers. The adults will still be nearby. At stage 4, you will see chicks that are fully feathered and ready to fledge and leave the nest. For the species that you will be tracking, you might encounter very large colonies of birds. In order to help keep track of the individuals and behaviors, it can be helpful to divide the colony into subcolonies. In this case, a barrier could help establish how you divide the colony. You can label one side A and the other side B. Or the birds could be nesting on different islands, each of which could be its own subcolony. This is what the first page of the datasheet will look like. In a complete datasheet, the whole colony name is written, a full date is used, full observer names are recorded, military time is used for start and end times, and the reason for poor visibility is recorded if the colony was difficult to see. The second section will be to write the total nest, adult, and young count, and note any possible nests you weren't sure about. A complete datasheet will have zeros filled in indicating that you were surveying for those species and did not see them. Blank spots leave room for assumptions and misinformation. This is the second datasheet you will fill out. This top portion describes the specific information for each stage that you can refer to when you are in the field. In this complete sheet, they named each subcolony, counted the total number of adults, and provided the appropriate count for nests and chicks in each stage. For all stage 0 nests, record the total number of adults. For all stage 1 nests, record the total number of nests. For all stage 2 to 4 nests, record the total number of chicks. The final portion of the datasheet has a section to include more information about how you defined each subcolony. It is essential that you fill this portion out the first time you designate a subcolony, but it does not need to be filled out every time after you first describe them. If you add a subcolony later in the season, be sure to record it in this section. We also ask that you attach a site map with the subcolonies shown to the back of the sheet. The chick in this photo is still covered in downy feathers, which makes it stage 2. Based on the proximity of the adults, we can assume that they are the breeding pair associated with this chick. We record them under the total number of adults column, but not in the stage 0 column, because they are beyond the pre-nesting stage. In a larger colony, you will likely have chicks at a variety of development stages, and will have counts for multiple stages in a given visit.